shortly. And thank you again for being here. I guess we met last February. I guess it was February was last time we met. So we've got things underway. We're excited to give you an update today. In the room here, we have I'm Brandy Keel-Reese, and I guess we can go around and say who else here. This is the team that we've now assembled. We've got Dr. Ruth Carmichael, Dr. Sinead Mihine, Anya Brown, Penny Dimitriades, and also Dakota L. Bilbrey. So we're going to let the students give their updates. They've all been doing great things as well, and we'd like to talk a little bit about where we're going from here, and also how we can get your help in a part of this project as well. So I'm going to go ahead and share my slides here. Let's see. Can everyone see that okay? Yep. All right. Thanks for the thumbs up. That's great. Okay. This meeting is being recorded just for posterity, and we'll post it online on the Wastewater Footprint website. And if you have any questions as we go along, please don't hesitate to just go ahead and unmute yourself and chime in if you'd like us to stop or repeat or just have something to say or comment. I won't be able to see your faces or hand raised, so feel free to just let us know if you have any questions. I'll monitor the chat. Oh, perfect. Yes, Dr. Carmichael will monitor the chat for us, so feel free to use the chat too if you feel inclined. Okay. There. Oops, sorry. I just want to make sure I wasn't talking over anybody. All right. Yes. Oh yeah, we do have some new team members that are going to that are joining us today as well. But if everyone can just take a quick moment to just give us your name and who you're with, and maybe your role with who you're with as well. And I can call it out, or you can just go ahead and start.
Hi, everybody. I'm Casey Calloway. I am the Chief Resilience Officer for the City of Mobile and excited to listen in.
Thanks for joining us, Casey.
Good morning, it's Rusty Grice. I'm with Auburn University Shellfish Lab with funding from Mississippi Alabama Sea Grant Consortium, and I'm a aquaculture business specialist.
Thank you. All right, I see Cade.
Hey y'all, my name is Cade Kistler. I am the mobile baykeeper, and we've been lucky enough to have a role participating in this project and doing a little extra source tracking in the Dog River watershed.
And so Cassie's gonna jump in there too.
Yeah, Cassie Bates. I'm the program coordinator with Mobile Baykeeper, and like Cade said, we've really enjoyed going out and contributing to some data collection with this project.
Thank you. I see Katie next on the screen.
Hi, I'm Katie Falter. I'm with the Nature Conservancy, and we are just excited to be added to the group here.
Mimi?
Yeah, this is Mimi Fern. I'm with Dog River Clearwater Revival. We've got a pretty active monitoring program doing bacteria as well, so we're interested in this project.
Okay. And Will?
Good morning. This is Will Underwood with the Maryland Department of Conservation and Natural Resources State Lands Division. I'm our Coastal Section Administrator and also involved with Weeks Bay Reserve.
Excellent. All right, am I missing? Bill Burkert. Up there. That's fine. There we go.
Hi, it's Bill Burkhardt. I'm the director of FDA's Gulf Coast Seafood Laboratory here on Dolphin Island.
Thank you. Glad you're here. See Scott?
Hi, this is Scott Reichert. I'm with the Auburn University Shellfish Lab. Do a lot of work in the hatchery and work with a lot of oyster farmers. So all this is of interest to us.
Glad you're here. We're going to talk a little bit about oysters at the end today. Elizabeth?
Hi, I'm Elizabeth Heeb. I'm a research technician in Dr. Carmichael's lab at the Dauphin Island Sea Lab.
Thank you. Aisha?
Hi, excuse me, I'm Aisha Gray. I'm the director of the Grand Bay Mirror, and we also have a research project that's similar to this one.
Fantastic. Did I leave anyone off? Okay. Chuck Wilson, are you on? Maybe a connection issue. And then Carl. Oh, Carl, yes, thank you. Oh, Carl, you're muted.
All right. I'm Carl. I'm a researcher at Dauphin Island Sea Lab in Dr. Carmichael's lab. And I do a lot of research pretty broadly on like trophic and movement ecology.
Fantastic. Thanks to everyone for being here. If I missed anybody, feel free to jump in there, but I think I got them all.
And we have several people who are on who are new, and so thank you guys for introducing yourselves. But you're not necessarily new in terms of your organization was part of our project since the proposal stage. But we do have two potential new participants, the folks from the Nature Conservancy, which I'm really excited you guys are able to make it. And so we've highlighted you there in red because it's a whole new partner. And then also we're working on getting a partner from the Water Institute of the Gulf. And I didn't see her on today, but hopefully we'll be building on that moving forward. So if any of you all have ideas about other folks who you think would benefit from this information, or other groups that we should be working with, please feel free to just shoot us an email after this or even during this or put it in the chat, and we'll make sure to reach out and make those connections.
All right, and just to guide you, you can see that we've bolded those in red, the Nature Conservancy and the Water Institute of the Gulf. That is deliberate, that you'll see that a lot of what we're showing, we showed last time as well on some of these slides, but we've highlighted in red anything that is new or that is different or that has been updated so you'll be able to see it quickly. All right. And again, thank you to everyone that has enabled this research. This has really been, an amazing group that has been assembled, and all with the common goal of, understanding microbial pathogens to our waterways. Okay. To review our objectives, this was what was stated in the proposal, and I'll go ahead and just read those out. But we're looking at defining the microbial sources to the Alabama waters. We want to distinguish at least wastewater treatment plant versus septic wildlife. That's including waterfowl and wild boar. We have some others that we've added since the proposal was written, and we'll give those updates as well, livestock and other non-human inputs. We want to define the conditions, the location and timing of microbe influences. We want to use, the ancillary physical parameters with, combined with these geochemical parameters, combined with these microbial indicators to try to start to conceptually model or diagram, the conditions in which we have, microbial pathogens could be highest or problematic. We're facilitating data sharing using our wastewater footprint, which was put together by Dr. Ruth Carmichael as a part of other water quality research that she has been doing. And so we're using that great website infrastructure to share data that we have coalesced, and we're going to talk about how we're going to need your help and the help of other folks that aren't here today to help us build up this this metadata or access to this metadata to try to find it in one quick place in all at the same time or all in the same place. We've also are wanting to enhance local capacity for microbial source tracking, where part of what we're doing is using traditional markers, the traditional tools of the classic microbiology tools of plating and plate counts and growth, and then also combining that with some more advanced techniques like quantitative PCR, real-time PCR, and then also environmental DNA. So looking at the entire community of microbes and also any of the signatures that animals might leave behind as well, using high throughput sequencing. But we want to bring that to the stakeholders, bring it to you guys, and also to others by making a toolkit that others can use and apply this high throughput sequencing in their own labs or in their own tracking efforts. And this is just again a background of what we're trying to combine: the classical, the advanced, and also using the biogeochemical parameters as another way to track wastewater inputs through stable isotopes and nutrients. So we're really trying to link these existing tools and these data sets together in a more conceptual and statistical way. All right, our sites have been updated just slightly, and thanks to everyone's inputs. We have been very grateful to get everyone's help to, make sure that we refine these sites. We've worked with the FDA to refine them a bit. We've also worked with Mobile Baykeeper to, add sites to this. So it's been a great effort. What we have here is just that same map that you've seen before, either from the proposal or from our first stakeholder meeting. And this is our, these are the sites that we have begun sampling at. So we're excited to report some of those efforts. And I'm going to turn it over to Anya Brown, who is a graduate student that is working on, a part of this project. The arrow works.
Hi, everybody. My name is Anya Brown, and as Dr. Keeler mentioned, I'm a PhD student in the Carmichael lab. I'm involved with the field sampling as well as evaluating some of those classical microbial indicators and the biogeochemical indicators. And so the goal of this project is to sample monthly all of these sites on the outgoing tide for the entirety of a year. So we started back in September. We have done already 2 sampling events, and we're gearing up for our 3rd sampling event this week. And we'll proceed to sample until the end of August 2023. So the way that we handle this is that every month we split the sampling up into 2 days, where we handle all of the western portion of the bay on one day, and then the eastern portion of the bay on the next day, just to manage those sample processing time. So when we go out, you can see in the lower left corner, those sites 1 through 4, those are actually on water sites. So we take our boat out and sample each of those sites. And you can see we've had some nice weather up until now. And then we get back on land and proceed north to head to the rest of those western sites, including the wastewater treatment plant in Mobile. After we do that sampling, we head back to the University of South Alabama campus, where we proceed to process those samples from the day. The next day, we visit the Dolphin Island site, and this was just to manage sample numbers per day before heading over to the east coast of the bay, and then proceed north and around the bay on the east side. And then similarly, we end up at the University of South Alabama to process those samples from that day. For the classical microbial indicators, we have a new lab at the South Alabama campus specifically for this work. And you can see I've been doing this work with the fecal coliforms, E. coli, and male-specific coliphage here. For fecal coliforms and E. coli, I use a modified MTEC method where you filter that water through a membrane and plate it on those plates you see on the lower left. And then for the male-specific coliphage, we use a double layer agar assay technique. And you can see that on the right-hand side. I'm reading those results there. And then another fraction of the water samples is used for the advanced microbial source tracking. And I will hand it over to Penny to talk a bit more about these samples.
So, hi everyone, I'm Penny. I'm basically Anya's counterpart for the graduate students. So I am Dr. Killius's new PhD student, and I'm responsible for the field work as well as the advanced microbial source tracking portion. So I just wanted to give everyone an update of our progress with the advanced part. And I thought a good way to do this would be to provide a flow through of our process. So Anya just mentioned our field sampling and our progress with that. So once we take those samples back into the lab, I filter them to collect all of those cells on a membrane. And what I can do with that is then extract the DNA from that. And that extracted DNA is then what we use for the environmental DNA aspect, which is looking at all of the organisms in that water sample or that have come into contact with that water sample, as well as looking at specific organisms, which is the qPCR aspect. And that is where we are right now. We are working on actually validating our primers. So we want to make sure that when a pig pops up in our sample, it's actually a pig. So we're working with positive controls. We have collected fecal matter, and thank you to Grand Bay for giving us feral hog intestines. That picture right there is a gel that we made. It's actually our pig primers with the feral hog intestine, and that is exactly what we want to see. So our primer validation is going very well. And once we finish with the rest of our species, then we move on to the actual qPCR. So good things in the advanced microbial source tracking world. Then I'll pass it off. I'm not sure.
Yeah, that's all right. So yeah, we're making some progress. We've got 2 sampling trips down, and we have, let's see, I guess it would be 11 more to go. So we're, going to gear up for an exciting year. We are also going to, in addition to this, we're, collecting, of course, other water quality data and other metadata that will help, discern some of this, the molecular and the microbiological data. We are going to incorporate 4 or 5 sites for sediment. We have 4 sites that were laid out, and a 5th possible site as well. That is outside the scope of this project, but it is within my wheelhouse of microbial ecology within the sediments. Is something that I've done for the past over 15 years. So I think it's important to kind of dovetail a few sediment sites in here, so we can try to parse out the influence of sediment on these on some of these pathogens as being a potential source or sink of them. And let's see, a part of this too, we were able to, I'm going to let Sinead mention some of the work that another aspect that we've been able to add on here, which was Dog River, which is something that wasn't in the original plan, but thanks to partnerships, it's been really useful.
Yeah, so
We partnered with Baykeeper and we went out on 2 sampling events with them, sampled a number of sites all along the Dog River. And so they've taken some their usual data on kind of the fecal indicator bacteria, and we're going to be doing qPCR microbial source tracking on all of those. So as Penny said, in relation to the main project, right, we're validating our primers, making sure that they work in this context. But we have the DNA ready to go once we've got the final primer sets validated. So we're very excited because that's going to give us a much different sampling scale in terms of a detailed look at one of the inputs into the bay that we can contrast then with our larger scale picture.
Fantastic, thank you. All right, and for this for our third objective, we want to, collect the studies and all in the same place, right? We all have the same mission. We all are doing, microbial source tracking, but there's also, other water quality, data and, ancillary data that is very disparate. It's in different repositories, different databases currently, hidden among publications, and sometimes in people's filing cabinets, So what we're trying to do is put all of that together in a single place where, and not necessarily the data itself, but the information about, the data, the papers, the databases, what indicators are present. And so I'm going to let Dakota, who has been amazing at leading this, she started working with us this summer and has been, our data manager in all of this. And so she's going to kind of introduce what we're doing with that platform.
Hello, everyone. My name is Dakota. I'm a research technician, and like Brandy said, this has been my primary role in the project, to catalog and record and organize all this data that I have been digging around through contacting agencies or just through Google Scholar searches. So as Brandy mentioned, our goal is not to necessarily collect all the actual data itself, but more so guide users to where they can potentially find all this information into a giant resource clearinghouse that we're going to have on our wastewater footprint website. Brandy, would you mind clicking that on the last slide to the website?
Well, let's make sure we're not sharing the right thing. So we need to stop sharing and reshare.
Okay. New share and this one.
Yeah, well, you might want to do the one that shows both so she can go back and forth.
Okay.
So maybe that's the
Screen, I think. There. Perfect.
Yeah, awesome.
Okay, great. Thank you. Good call. All right.
So, as most of us are familiar with, here's the wastewater footprint website put together by Roots Lab. The new addition to this website is this tab right here, specific for this project, Alabama Microbial Source Tracking, or MST. And by clicking this link, we're taken to the main web page of this project that describes a brief overview of what we're accomplishing and what we hope users will gain out of this, which is a data avenue where users can go through and look for data resources that may pertain for their future monitoring efforts or other projects. And the actual metadata itself can be found by clicking this link. So this is our current metadata clearinghouse with information that contains things like organization name, data availability. Probably the most key aspect is the related publications or data citations, and this will serve as the guide that users can follow if they're trying to locate a particular data set, as well as the information that Brandy previously mentioned with things like indicators or source tracking data that are used, start date and end date as well. And we also have some secondary information here in this button right here. So users can toggle through and select that information if that is something that they'd like to look at. And so initially, this is all information that I have gathered myself just through individual searching. Like I said, whether it was a Google Scholar search, which is where I found most of the research papers that we've included, or if it was contacting agencies and asking information about their data that they've collected over the years. That has gone on pretty well, but Brandy, Ruth, and I kind of sat down and put our brains together to think, okay, how can we increase this effort? There's only so much that one person can do through their own initial contributions. So this is what led to the development of our metadata questionnaire, because we thought about it and we discussed it and came to this conclusion that this is going to be a great way to encourage contributions from other potential users in the area that maybe we haven't included, or maybe might be interested in seeing what we've got cooking. So I'll take us to this link. Oops. To our oh well, I can fix that. So. I went through this, I practiced this, so I was ready. All right, now we're cooking. This questionnaire is also now available online as well by clicking this link. But just for viewing purposes, we'll go through it this way so we can look at the entire overview. So at the beginning of the questionnaire, we again express our project interests and our intentions with it, as well as discuss the goals that users can expect after contributing their metadata to this project. So we'll take a little walk through at this point. I'll be as brief as I can to be cognizant of everyone's time. Going through, we ask for basic background information, but coming up here on section 3, this is something, this is a keynote that I wanted to bring up. So we do encourage contributions from anyone that is willing to participate, whether it's research groups, monitoring organizations, but we are aware that some groups may have data that are not ready to be shared with the public yet, but we have an option here for users to indicate their data availability, whether it's publicly available, anybody can go and find it using a link or a DOI that will be provided, if it's available through request, whether it's contacting someone, or if your data are not ready to be publicly shared, we do have this option as well for users to at least submit their preliminary information and then possibly come back later on once it's ready to be shared and submit an updated response, or contacting us if they prefer that method too. So we at least give this option for like, hey, like it may not be ready yet, but come back next time and update it, and we're glad to give up there for them. Related publications and data citations. This is probably the main portion that is holding this questionnaire up. This is where we will collect citation information, links to data sets, and other ways that we can, that users can possibly use to obtain the information they're looking for. But as a reminder to our users and participants when filling out this questionnaire, this is only in relation to one discrete data set. The idea is one response is going to correspond to one discrete data set. So as many citations or links that apply to that one individual data set, they will be put here. And if a user has multiple data sets they'd like to contribute, we have an option for them at the end to submit another response that corresponds with that other data set. Going through, you'll fill out some basic background information and who is the owner of it, what was the purpose of the data. And this option here, which is one that we really wanted to hit home when we were drafting this questionnaire. We understand, and we again want to be cognizant of users' time. So we understand that maybe some individuals may have multiple data sets and they don't have the time to sit down and fill out all the metadata that we were asking of them. So we do have an option here where users can enter the metadata themselves manually by filling out the rest of this survey, or by just submitting the links that we asked up here or the citations. we'll have our source tracking team use those resources and fill out the metadata for the user. And that'll probably be my responsibility as we get submissions coming through. I'll use the resources that are presented to me to go and fill out that metadata if that is the option that a user selects.
And Dakota, at any time, so for example, if somebody selects, yeah, I'm going to give you my basic information and here are all of my papers from this data set, and they select to have Dakota enter the metadata, at any point you can go through and check it. And when she's done, she'll communicate with you and you can go back and see that file that she showed at the beginning, which showed everything she compiled. Even if she already compiled something for you, that gives you the opportunity to freely go in there, take a peek at what's in the database and make sure that we did it right. And so if there's anything that you want to change or update, you can give that feedback and Dakota can go in and update that and correct it. So you'll get a chance to also just have us do the work, but then you get to check it and make sure it's correct.
Absolutely. And that can be done also by just sending an email to us as well, just saying, hey, this probably needs a little bit of touch up. Thank you for bringing that up, Ruth. Let's say, for example, that you did have the time to sit down and fill out all the metadata information, and the user will then be taken through each section where we ask all the information that we're looking for compiling into this clearinghouse. So like traditional indicators and source tracking data used, have a various list of options available that users can select as many as apply, or if there's maybe something that we've missed or have not thought about including, user will have the option to enter anything else that maybe we didn't quite catch the first time around. Same thing with methods, detection that we use. The user will have the ability to select all that applies or enter another option. Sampling medium will also give the user the option to specify if they want to make a specific reference to a medium that was used. Where does the data correspond to? What's the water body? Are GPS coordinates available with the data? Is that something that would need to be requested? Sampling time period and frequency of your data. We've shortened it down to just giving us a year when it was done, or if it's ongoing, just to include present. The sampling frequency with the option to also again include an example that maybe we didn't cover. Project status, if it's ongoing or if it's a completed project. And we also have this option as well for those who possibly use multiple indicators or have different dates. We have this option here for users to select like, yes, I have a specification I'd like to include, or I don't, this doesn't apply to me. This is where the sampling frequency or sampling date specification will be noted. Like this, for example, let's say you have these two indicators that were used during this time period, but maybe you have a third one that has a different date range, or maybe it was following a different sampling frequency protocol. Those specifications can be made in this section. Does your data have supporting environmental data? Yes or no, pretty simple. If you do have it, please list it here in this text box. And then closing out, this is where we'll ask users if there's any organizations or groups that need to be acknowledged for their data. They can list that here below. Upon completing this questionnaire, the user will then be presented with this confirmation message where we express our gratitude for taking the survey, and we also provide links back to the Metadata Clearinghouse for the user to see other responses that we've collected so far. And here's a big point that we plan to drive home. As users submit this questionnaire, we want to encourage a sharing among colleagues and community partners. So we do encourage our users that after completing this questionnaire to share it amongst their colleagues and get the word out there, because we want to compile it all. As well as other links such as editing a response, submitting another response, or just our basic contact information to get back in touch with us. Okay.
Bottom right on present.
Here?
Yes.
Okay. So those responses will be collected in, I wonder if this works. Oh, well. So the questionnaire responses will be recorded into a private spreadsheet for us. in a Google spreadsheet like there, and each column will correspond to the questions that we've asked in the survey. And those responses will remain in there. And as our team is going through it, again, this will probably mostly be my task. As the responses are collected, we will review them and kind of filter them out and format them to where they will fit the public clearinghouse that I've shown you already. So basically, to think about it in simple terms, it's going to stay private until it's been tidied up and formatted to fit the public spreadsheet. And so again, that point that I mentioned earlier, we will gladly be inviting contributions to this clearinghouse, using the resources on our website, as well as drafting an invitation email that will have all the links that I've shown you already to the clearinghouse and to the wastewater website. And again, encouraging the sharing among colleagues following the questionnaire submission. So yes, we are gladly encouraging metadata sharing among our partners through this questionnaire and other resources available. And he's not here today with us, but big thank you to Pat David, our data architect at Thistle. Without his expertise and his help, none of those websites would have been made possible. So thank him very much for his time on this. He's been great to work with, and I'm glad to see how the website has looked now.
Equally, I'd like to thank Dakota for, starting from scratch and building, this wonderful repository of all of this, metadata about these papers. And she has found, a lot so far on her own. But again, it's really important that we get you guys to, help check it, but also to help contribute or let us know who should be helping contribute as well.
And I also want to point out that Dakota crafted that survey too, which I'm sure she could tell you as we started going into it, Brandy and I, and she made like, oh, and one more thing, and oh, we should add this, and oh, we should add this, and it got bigger and bigger. But I think as a result, the product is really thorough, but yet as efficient as possible, because we're trying to make it not so labor intensive for the folks who are contributing, which means it's been more labor intensive for Dakota. And so she stepped up and done a great job with that. And you all can expect to see an email from her/us with this survey in it, and all the things that she showed you, so you can poke around in the clearinghouse, and you can also contribute data, but then also, like Dakota said, share it around to all of your other colleagues who might have data. And then one other final point, while we're doing this for the purposes of informing microbial source tracking, and that's our project title, we recognize that people who are collecting this data originally may not have been thinking about it in those terms. So we just also want to remind everybody that if you have any kind of data that fit into this that can be put together, you might be thinking to yourself, well, I didn't collect fecal coliform data for the purpose of source tracking, but we can potentially use it in that way now. If that's the power of us all combining our data together and making it available. And so just encourage you, regardless of what your original intent was for the data or what your colleague's intent was for the data, we still want any of those kinds of data, if that hopefully makes sense.
I'm going to pause here in case there are questions. That was a lot that to process, and that's a big part of what we've been pushing in this first part. Of course, getting our sampling program underway has been exciting, but also this metadata clearinghouse. If there's any questions, we'd all be glad to answer those right now. Well, good. Well, then we do look forward to everyone contributing though too. And so checking us. The end goal to all of that would be that if it's somewhat in the scope of this proposal as well, but and forward thinking that we can model some of this data, that we can having these long-term data sets are amazing to be able to model and make predictions from. So that's what we're excited to see, into the future. But for the first step, we need to know that these data exist and what these data entail. And then this is, something that we're going to be working on in the future. I mentioned this at the beginning about the eDNA toolkit. We're working on this for years 2 and 3. So, although we're formulating ideas about it right now, we haven't begun to assemble this yet. And what I mean by toolkit, we intend on having, those virtual and hopefully live and in person, workshops to train users on using eDNA and taking us, into the next steps, even beyond qPCR. And eventually that will be hosted on the DISL website. But for now, stay tuned. That's something to look for in the next couple of years. About moving forward. Okay. Making sure. So we did start on our September was our first sampling date. That was intended to be a practice run, but it went so well that that's going to be part of the data set. So we are going to have 13, instead of just 12, we had one for a practice run and then 12 that were budgeted for. And so we're going to end up having 13 great sampling time points. What's going on now is still compiling the existing data for that metadata clearinghouse that we just described. We are going to continue our field work. Rest of this year, which is 2 more sampling points, November and December, and then into 2023. We are going to be leveraging some sediment sampling through my lab, and then also some oyster sampling. That's something that we're aiming to do. Both Penny and Anya have been excited to incorporate these oysters into this. So we're going to set out some oyster traps, working with the FDA on that, and ideally placing those where the sediment sampling is going to be located. So we'll have water data, sediment data, and of course some oysters that are going to grow out for a month or 2. And then we're going to analyze those for some of those same microbial indicators that were incorporated into this project. I'm going to continue with meeting with you guys biannually, but between the meetings, please feel free to reach out to us. Feel free to, give us ideas if you want to partner in some other way. We don't have to wait till the working group meeting until these, stakeholder meetings. Feel free to reach out to us anytime. And then look for that eDNA toolkit workshop in work 2024.
Please. I also wanted to just mention briefly, as part of the sort of new leverage work that we're doing with sediment and oysters, this is the collaboration with our partners on this project and through the joint program with the DISL. It's a collaboration with the U.S. Food and Drug Administration, who has the Seafood Safety Lab on Dauphin Island. And certainly, while we didn't originally write sediment and oyster sampling into the EPA-funded project because there was just only so many resources and so many sites, the cool thing is with this collaboration with the FDA, we're able to add some of these components. And certainly from the beginning, this project, our intended application was for thinking about seafood safety and public health as well. And we had our shellfish farm sites looped into and important harvest areas already looped into the water quality sampling for this project. So it's really a natural thing once we were able to secure the funding and be able to do that to add the actual oyster sampling as part of this. And so we're just very grateful to also our FDA partners for making that part of the project, allowing us to do that and make that part of the project moving forward.
Fantastic. All right, I believe that is, yep. Just again, getting involved. Thank you for attending today's meeting. We really appreciate you taking the time. And I know we're kind of getting close to the hour, so I want to open this up to questions. Again, feel free to give us feedback. we really want to make this clearinghouse something that's a powerful tool for now and in the future as we want to, get towards modeling these data or, conceptualizing these long-term data sets. And I believe, yep, there we go. If you have any questions, we're glad to take them now.
And you also can ask questions to the students too. They'd love to get a chance to tell you more about their work.
I've got one that's really just logistical. Will y'all please send out the survey link after this call so we can have easy access? Perfect, thank you.
Absolutely. This meeting will also, that's been recorded, will be posted on the wastewater, our wastewater footprint, sorry, just went blank, and also the slides, but we'll make sure that you have a live link as well.
And it might not be immediately after this meeting that the survey goes out, but it will be soon. We have a couple of tweaky things I think left to do. We need to do an actual test run, but we will definitely circulate that to we've got you. We've trapped you. We are going to circulate that to you for sure. So it will be coming soon.
I have a question for Dakota. I know we talked early on about the Alabama Water Watch citizen science data, and I'm wondering if you figured out a way to incorporate any of that. I think it's important for us to engage our just residents of the area and contributing to the data sets.
Yes, so I was able to add the Alabama Water or AWW's data into that clearinghouse, mostly for the Mobile Bay area. I know there's other data that has been collected for the more northern part of the state, but I have added that to our clearinghouse with all the metadata information that I could compile, also based on our conversations that we had via email earlier in the year, which has just been following the resources that I can on the website and matching the information I've been finding there to fit the format of the spreadsheet.
Okay, that's great. I know some of those data sets go back at least 10, almost 20 years on some of them.
You were one of our early guinea pigs essentially on this. I know you may not realize that, but because your data set was one of the first ones that Dakota started working on, it was really one of the first ones that helped to shape what this database would look like. So we thank you for your contribution.
Absolutely.
If there's a question. Oh, yeah. Thank you. Well, if you think of any questions, feel free to reach out. We really appreciate your time, and we look forward to moving this forward and working with you all.
And we'll hang on here for a minute or so. If anybody has a question you want to ask in private or whatever, feel free, but we're here. You have us all in one room, so feel free to hang on and chat with us too if you'd like to. Are we done with everybody else?
I think so.
And so we did a great job keeping it under an hour.
Thank you, everyone. Thank you for your time.
Thanks, y'all.
Thank you.